On behalf of the NCI team here, I'm really pleased to be able to announce the launch of the NCI Serological Sciences Network, or as James said, SeroNet, and to announce the grants and contracts that are being awarded to form this network. And this network is going to be a central element, of the NCI's ongoing response to the COVID-19 pandemic. As we all know, earlier this year, our nation's scientific community was called upon to address COVID-19 worldwide pandemic. NCI is one of many federal agencies with the essential expertise and the resources from Congress to respond to this unprecedented challenge and rapidly deploy both funding and scientific leadership. In April 2020, Congress provided $306 million in supplemental funding for NCI to study serological sciences related to COVID-19, specifically to mobilize the U.S. research community to better understand the immune response to the coronavirus SARS-CoV-2, which is the virus that causes the COVID-19 disease. While, of course, cancer and cancer research and cancer care remain NCI's primary focus, we're supporting COVID-19 research because of NCI's deep expertise in virology and immunology research, including research on viruses that cause cancer, such as HPV, and our experience in immunotherapy. Could I have the next slide, please? The overarching goal of SeroNet that we're launching today is to, number one, increase our national capacity for serological testing, while number two, also increasing our understanding of all aspects of the immune response to SARS-CoV-2. The research that will be conducted through SeroNet should begin to answer fundamental questions expected to have significant public health impact. Among those questions are, why do some people who are exposed to COVID-19 get sick while others don't? Why do some people who test positive for COVID-19 have severe symptoms while others have much milder symptoms? What kind of immune response is necessary to gain immunity from vaccination versus from a prior infection? Does disease severity correlate with long-term immunity to reinfection? Do people with health conditions such as cancer or diabetes or heart disease have worse outcomes in both the short term and the long term? And what is the prevalence of SARS-CoV-2 infection in the US across age groups, racial ethnic groups, and urban-rural populations? With today's launch of SeroNet, 25 of the nation's top academic and scientific research institutions will partner with NCI and the Frederick National Lab to help us answer those questions as quickly and accurately as possible. Next slide, please. I want to spend just a few words on the immune response and its relationship to serology. There are three basic components to the immune response, all of which contribute to how a person responds to an infection. The innate response is a person's first line of defense against any infection. It's not a specific response. That's followed by the so-called adaptive response, which is tailored to a specific virus. You've probably all heard of T cells and B cells. So T cells are involved in cell-mediated immunity and can kill infected cells and mediate long-term immunity. B cells are responsible for humoral immunity, that is, making the antibodies that are in your blood and in your serum. Now, let me just stress that serology is only a part of the immune response. It measures a person's immune response to an infection by measuring the antibodies circulating in the blood against the specific virus. Serological testing helps identify who has been infected and the seroprevalence within the population. Again, serology is distinct from the diagnostic COVID-19 testing that's been in the news as the pandemic unfolded. Serology has important implications for how we understand SARS-CoV-2, but should not be confused with the majority of the testing taking place to identify currently infected individuals. Doug Lowy is going to talk more about serology in his comments in a few minutes. Could I have the next slide, please? Within the overarching framework of SERNet's goals, its specific objectives are to A, develop novel serological assays and importantly, deploy them in the community. B, to characterize the biology underlying the innate, cell-mediated, and humoral responses to SARS-CoV-2 infection. C, determine the factors, whether genetic or environmental, that modulate the immune response and disease severity. And D, identify and remediate barriers to access to serological testing. In developing SERNet, we've worked closely with NIAID and will continue to engage with FDA, CDC, BARDA, NIST, and other agencies as well going forward. Of note, SERNet is collaborating with other research programs, including the NIH's All of Us, to test serum samples from, from more than 40,000 participants who've provided blood samples between November 2019 and March 2020. This collaboration will test for the prevalence of SARS-CoV-2 infection in the American public in the early days of the pandemic. And lessons learned from SERNet research could be applied immediately and may prove valuable to public health beyond the current pandemic. Next slide, please. Turning to SERNet's organization, it has 5 components. The Serology Lab, which is housed at NCI's Frederick National Lab, is among many activities working with the FDA and other government agencies to evaluate commercially available serology tests for COVID-2. The SERNet Coordinating Center at the Frederick National Lab will coordinate and foster collaboration across all of the SERNet components so that the network will function as an interactive and collaborative unit, with each component pursuing their own work, but also collaborating across the network and contributing to the overall goals of the network. Critical to SERNet are the other 25 partners that make up the remaining 3 components: 8 centers of excellence, 13 research projects, and 4 capacity building centers. All of the awards that are being made were based upon scientific merit as determined by peer review scores and program priorities. Taken together, the awards represent the breadth of research topic areas called out in the funding opportunities published back in June. In addition, we considered the geographic diversity of the research sites, as well as racial and socioeconomic diversity of the populations that were proposed for study in the research plans. In the next 2 slides, I'll summarize the grants that have been funded. If I could have the next slide, please. The 8 SERNet Centers of Excellence. Next slide, please. The Centers of Excellence, there they are, will conduct research to characterize the immune response to the coronavirus infections and understand what drives the immune response, disease progression, and protection against future infection. These grants are funded through what's called the U54 mechanism, which encourages multiple coordinated research projects at each awardee institution. These grants often have a shared administrative core and in some cases other resources to be shared across the center. Award sizes for these grants range from about $815,000 per year to $2 million each year for the 5-year duration of the award. Next slide, please. SERNet is also supporting 13 individual research projects at 12 institutions to conduct research that's similar to that of the Centers of Excellence, but with a more focused approach. These research projects are smaller in scale and scope. Typically, they'll be carried out in a single lab. Here, the award size is also smaller, ranging from about $500,000 to $750,000 each year for 5 years. Next slide, please. And the final component of CIRANET are the capacity building centers, which have been awarded through 4 subcontracts through the Frederick National Lab. These centers are charged with developing and expanding serological testing capacity and practice in the community. They'll scale up screening capacity to reach at least 5,000 samples per week with assays that have been granted FDA emergency use authorization. These centers are also going to be performing long-term studies of the prevalence of COVID-19 across the country. Next slide, please. It's important to note that all of the CIRANET components will work closely together, sharing information and resources. Furthermore, all of the publications funded through CIRANET will be published in an open access format and the underlying data made immediately available. Through the coordinating center, all of the samples and data generated by CIRANET will be made publicly available. Could I have the next slide, please? So with the supplemental funds provided by Congress, NIH is also leading or partnering in some of the activities listed here. And you can learn more about these and other activities on our website, cancer.gov, and connecting with the NCI press officers, and James gave you the email earlier. I'm now going to turn things over to Dr. Doug Lelly, who's going to provide some more background on COVID-19 serology and what we know and have yet to learn, and tell you more about the serology dashboard, which is coming soon.
Dr. Singer, thanks. And I want to welcome everyone on behalf of the NCI. And I'm going to talk for about 10 minutes, really with the main goal of trying to help you understand a little bit more about what NCI has been doing in the area of serology, but also to try to help you to understand more about what serology is, and as James Alexander mentioned, what it isn't. If I could have the next slide. This is a quote from Maura Judkis back in May, a journalist at the Washington Post, who said, "No one wants to have COVID-19, which is a disease caused by SARS-CoV-2 infection," as Dr. Singer mentioned, but everyone wants to have had it. And a large part of what serology is about is to try to verify whether the degree to which this statement or the underlying understanding of this statement is correct. And I don't think we have good answers right now, but I am hopeful that by the end of this year that we will have at least partial answers, and I'll go through that. If I could have the next slide. This simply repeats for you one of Dr. Singer's slides, simply to remind you of what serology is and what it isn't. There are these 3 major components to the immune response: the innate immune response, the cell-mediated immune response, and the humoral immunity. Although all of this is made by cells, serology is really looking at antibodies, and the vast majority of the antibodies that people are looking at is antibodies in serum. Serum is blood that has coagulated, and the cellular part of the blood has been separated so that what you're left with is a clear fluid that is referred to as serum. Plasma is similar to serum, except that it's blood that has not been coagulated. But antibodies also can be measured in other fluids, for example, in saliva or in the nasal passages. But most of the research and the data that come out related to SARS-CoV-2 infection are antibodies from the serum or from the blood. Next slide, please. This slide goes over a number of the outstanding questions that we look forward to being addressed, at least in part, with the study of serology in people who have and have not been infected with SARS-CoV-2. Is being antibody positive associated with protection against reinfection? The hypothesis is that that's the case, but we don't yet have evidence to demonstrate that. If being antibody positive were associated with protection, what antibody levels may be associated? And again, we don't know that. And if there is protection, how long will protection last? For example, somebody was infected, they become antibody positive, but several months later, they become antibody negative. Does that mean that they no longer are protected? We simply don't know the answers. Another important question is, what about herd immunity? What seroprevalence levels are needed in a population for herd immunity? And the speculations go all over the map, but suffice it to say that we don't know. Seroprevalence is actually what percentage in a population have antibodies. NCI is funding research to address these and related questions through SeroNet as well as other research, and much of it is in collaboration with the National Institute of Allergy and Infectious Diseases and the Center for Disease Control. Next slide. This slide lists for you 3 additional NCI serology efforts in response to the COVID-19 epidemic. Starting in April, we began to characterize the performance of many serology devices submitted to the Food and Drug Administration. And we then submit our results to the FDA. And I'll have something to say about that in a couple of additional slides. This is a collaboration that we have worked closely with the FDA, but I should also point out that BARDA and NIAID and CDC have been critical to our ability to do this. In June, we started to develop a dashboard for the United States to provide information on antibody studies in the United States. And I'll have something to say about that. It is still under development, but we look forward to it becoming live in about another month. The third area is to develop a standard of SARS-CoV-2 serum that can be used by vaccine manufacturers and by institutions producing convalescent plasma or serum for treating COVID-19 patients. And this standard will enable valid comparisons either between vaccines in terms of their immunogenicity or for convalescent plasma and serum in terms of making sure that the antibody levels are what they are, and are they sufficiently high. Again, this is a work in progress that we just began in September, and we hope sometime next month to be able to have that standard. If I could have the next slide. This slide summarizes what we have done thus far in the collaboration with the Food and Drug Administration. We have evaluated and sent to the FDA more than 100 commercial serology devices that were submitted to the FDA. We have focused on the antibody category called IgG, although we also test IgM for those developers of devices that use IgM. But IgG is what most people refer to because they come up, those antibodies come up with SARS-CoV-2 just about as early as IgM, which is usually an early marker in other infections. But they tend to persist, the IgG, longer than the IgM, so that they usually can give you a more accurate indication of whether someone was previously infected, although for short-term use, IgM has many utilities. What we look at particularly is sensitivity and specificity. Sensitivity is the ability of these devices to detect true positives. And there has been a remarkable variation that in some devices that were submitted to FDA, they have been as low as 30%, but some have been as high as 100%. Specificity has varied from 87% to 100%. And specificity means that the device does not detect false positives. And these results then are sent to the FDA to help the FDA determine suitability for emergency use authorization. And the results are posted at the FDA website once they make that determination. Next slide. This slide goes over the importance of specificity at low rates of seroprevalence. And low rates of seroprevalence is what we currently have in most areas of the United States. If a test has 99% specificity and the seroprevalence rate is found to be 10%, it means that only 10% of the positives will be false positives, or 90% of the positives will be true positives. If there were 100 tests, 9 of them would be actual positives, and 1 will be a false positive. A 95% specificity sounds very good, but in that situation, if seroprevalence rate is found to be 10%, 50% or one half of the positives will actually be false positives. Again, for this hypothetical 100 tests, 5 of them will actually be positive and 5 will be false positive. So that when you look with a test that only has 95% specificity, you would overestimate the degree of the population that was previously infected. Next slide. So seroprevalence studies now can be used for the antibody studies can be used to measure seroprevalence. And these tests should be able to identify virtually everyone who has had symptomatic infection and most of those who have had asymptomatic infection. I want to repeat something that Dr. Singer mentioned. Antibody tests are not used for diagnosing current SARS-CoV-2 infection. Diagnostic tests instead identify parts of the actual virus, either viral RNA, which is most of what's used, or in some instances, measuring viral protein. Next slide. Developing a SARS-CoV-2 serology data and dashboard is something that we were asked to do by HHS, CDC, and NIAID back in June. And we're still developing this. But part of the reason for NCI being able to do it is the Frederick National Laboratory has expertise in developing dashboards actually through our clinical trials reporting system and other databases. The key features are to make this a publicly accessible data warehouse that would systematically document and track SARS-CoV-2 serology studies and associated test results, and then to have a tracking dashboard to visualize the serology data and to present the results overall and by key strata. I will show you a hypothetical mock-up in the next slide, please. And this is, as I mentioned, still a work in progress, but we hope that it will go live with actual data sometime next month. What there should be is a dynamic pop-ups that show any data elements that are deemed appropriate, and we can do this at the level of the state, county, or zip code, and also look at time trends. We have interacted with a group in Canada that is developing a serological dashboard that is analogous for the world. And our system is designed to be compatible and interoperable with theirs. But our system, as I mentioned, will give you data at the state, county, and zip code level and time trends, whereas the Canadian system is just showing things at the county level. Uh, I want to thank you very much for the opportunity to tell you a bit. And as shown in the next slide, this ends the presentations.